Well, here we are at the last key area that we need to cover for Unit 2. I bet you thought we'd never get there, but we are almost done. Last hurdle. So this key area is having a look at what sort of things affect the control of microorganisms or affect the metabolism of microorganisms. But this time round we're looking at genetic control of the metabolism of these organisms. In the last section, key area 2.6, we were looking at environmental conditions, the pH, the temperature and so on. Now what you have to think of first of all is that you're some sort of industrialist and you're looking for a useful product that a microorganism may produce. And so what actually happens, and some companies do this, they go out and they have a look at what microorganisms might be growing in, say, a soil sample, for example, and they'll take that soil sample and they'll grow various microbes on it, and they'll put the sample onto a petri dish. These microbes will grow and you'll get individual colonies eventually growing on there. They will then isolate out these individual colonies and have a look at them and see if they will produce useful substances. And what they do, in fact, is they grow these microorganisms on all sorts of different media, and they will culture them up and produce pure strains, and then they'll have a look and see if those bacteria produce any sort of useful product that can be used in an industrial process and again make lots of money. So here you are, you've done your work, you've isolated your bacteria that you're interested in, and lo and behold they produce a very useful product. But the actual strain that you've got, there might be some problems with it. It might be that they're not really very genetically stable. It might be that they actually cost an awful lot of money. The nutrient that you're using, or the nutrient agar that you're using, is a pretty expensive one. So you may want to improve the ability of that microorganism to grow on lower costing nutrients. You may want to improve it in order for it to produce more and more of the stuff that you want. And you maybe want to make some sort of improvement which makes it easy to harvest the compound that, or the whatever it is that you're producing, the useful substance, after the fermentation has been completed. So there's a number of reasons why you might want to make some sort of improvement to the microorganism that you have isolated. And there's these 3 ways that these improvements can be brought about, and we're going to talk about them as we move on in the video. The first one here is looking at this term here, mutagenesis. Or we could have a look at breeding programs, and we could all have a look at maybe genetically modifying the organism. We'll look at the genetic modification in a different video. So in this video here, we'll have a look at these 2 things, mutagenesis and breeding programs. So what is mutagenesis? Well, it's the creation of mutations, and that can be done by exposing your microorganism to some sort of mutagenic agent, something such as UV light, something that we're not seeing an awful lot of in this country at the moment, but we're going to have a look at this process in the next slide. Well, here you are here with your wild type of bacteria, and you're wanting to make some kind of change to it. What you want to do is you want to change it from growing in some sort of really expensive nutrient agar to a type of agar that is a lot cheaper. And at the moment, your bacteria that are in here have got very expensive taste. So what you want to do is try and create some sort of mutations that are there that are going to grow on this cheaper media. So you shine some UV light onto it, And then you take a sample of those bacteria and you grow them on the expensive media to start with. And so what you'll get is you'll get some of the colonies that are on that media will just be the non-mutated ones, but a few of them will have made some sort of mutations. And in this situation here, there's a wee sterile pad of velvet that's been produced. And what happens is that's pressed down on, well, we're calling it the master plate. That is this plate here. So that velvet pad is placed down on that. It's lifted up from the plate. And what happens is the cells have been transferred onto here. And then what they do is they can take that pad and they can press the pad down onto a plate that has got an agar plate that has got the cheaper agar in it, or nutrient agar. And hopefully that UV light that you've used at the very beginning has produced some mutant colonies or mutant organisms. And what they do is they then grow on this nutrient agar. They've got cheaper tastes. And the ones that you had up here that had the expensive taste, they were the non-mutated ones, they won't grow. And so what you can then do is, we'll just move this up a little bit. So what can then happen is, you then take this plate here and the one that you had away back up at the beginning, and you take the two of them and you compare the two of them, and from the comparison of the two, this is the one with the more complex media here, more medium, and this one here has got the cheaper medium in it. And what you can do is compare the two of them, and you're able to work out whereabouts your mutant colonies are. You can take them out and you can then grow them on the media for future use. Now, ideally what you're wanting to do is get some sort of improved characteristic there, such as some increased yield. However, sometimes mutant strains are genetically unstable. You are, after all, shining a whole lot of UV light onto them. So they have to just check that this improved strain that you've got, it's got to be monitored to ensure that it's still in its mutated state. You don't want to spend a whole lot of money growing it in a huge fermenter to then discover that it's actually reverted back to what it was originally, which is what sometimes can happen. So it has to be monitored and or tested quite a number of times to make sure that it isn't going to revert back to what you had originally. Now, if your UV mutations aren't working, then there are other ways of trying to change your microorganisms. One of them is looking at bacterial breeding programs. Now, bacteria reproduce asexually, and what happens is that new strains of bacteria can arise due to horizontal transfer of genetic information. If you remember before from our information, horizontal transfer in bacteria, if you're talking about sexual reproduction, is called vertical transfer of genetic material. And there's a number of ways that this can happen. You can either have bacteria taking up DNA fragments from their surroundings, or pieces or of chromosome DNA or plasmids are transferred from one strain to another. And there are different ways that can happen. Now, I've got this terminology here. I've got transformation, and you don't need to know these terms. I'm going to look at three things. I'm going to look at transformation, transduction and conjugation, but I've put the words in there just in case you come across the term. But this here, this transformation here, what is happening in this situation is that the bacterial genome is altered by transformation, and what's going on is that the cell takes up a piece of foreign DNA that is there in from the remains of a cell that's been destroyed and has undergone some sort of lysis, that is some sort of bursting. And some bacterial cells have even got proteins on their surface that are specialised for the uptake of pieces of naked DNA that have come from other species. So what we're seeing here is we've got a wee fragment of DNA from another bacteria that's got a different genome, and that DNA, or that foreign DNA, is able to enter into our bacterium, and the foreign DNA ends up replacing a part of the original DNA. It becomes incorporated within the chromosome, and so you end up with the original DNA that is degraded in some sort of way, and you end up with this change that is here. It's something that bacteria have developed over millennia, and because it's a way of, if you're thinking about the fact that they're reproducing asexually, there's not an awful lot of genetic change that's taking place there. So this here is one way of overcoming that variation that you get so much with sexual reproduction. It's not happening with asexual reproduction, but this is one way of overcoming it, because you're introducing some new DNA into your bacteria that may give some sort of advantage to that bacteria to help it survive. So that's transformation. Now here's another way that the genetic information in a bacterial cell can be changed, and it's this process here called transduction. Now we're looking here at a bacteriophage. That is a virus that infects bacteria, and what happens is a virus will infect a bacteria, say a previous bacteria, and when the bacteriophage attaches itself to the bacteria, it injects its own DNA in there, and when its own DNA is in there, then what happens is that DNA, all the machinery that is inside the bacteria for making proteins, kicks in, and lots of little bacteriophages are produced by the protein making machinery of the cell. Now occasionally what happens is that normally what would happen is that these bacteriophages would just contain their own DNA. There would have been copies of those made, and then these bacteriophages are then released from the cell. The cell is quite often it's killed. It bursts to release these new viruses, and inside them there is copies of the DNA that was in the original bacteriophage. But occasionally what happens is in this process is that a bit of the original bacterial DNA gets mixed up into the bacteriophage, so you end up with a bacteriophage with just a little bit of bacterial DNA. And then what happens is when that bacteriophage then comes up and attaches itself onto the surface of another bacteria, then there is some foreign DNA that is in there, and that foreign DNA, well this time round actually it is actually coloured red, that foreign DNA, and that foreign DNA that is there ends up being injected into the bacteria, and it is in this diagram here, and that foreign DNA aligns itself with the bacterial chromosome, and it ends up replacing the original DNA. It becomes incorporated into the chromosome, and the original DNA gets degraded. And so therefore you've got different DNA contained in here that has actually come from this bacteria here, and it's ended up in this bacteria, so you've made some sort of genetic change. Okay, here's the third way that genetic information can be changed in bacterial cells. So this is called conjugation, and what happens with this is here you've got two bacterial cells a donor cell and a recipient cell. And you'll see that there's something called a conjugation tube that it's a temporary bridge between two bacterial cells. These two bacterial cells have got different genomes, but what this bridge can do is that a plasmid can be passed from one of the cells to the other. In this case here, we've got this plasmid here, and it may actually have some useful information that could be passed on to this other bacterial cell. And this is one of the ways that, for example, maybe a gene that helps with antibiotic resistance that is contained in this plasmid, that information can be passed on by this process called conjugation to other bacteria cells that don't have that advantage. And so what happens is the copy of the genetic material or this plasmid is passed to the donor cell from the recipient cell. So you end up at the end of the process with two bacterial cells that have both got this plasmid in it and the useful gene that's contained within it. And now you've ended up with a cell that has got a new combination of genetic material in it. Once this conjugation tube, I forgot to mention that, it disintegrates, and then you end up with these two cells. Well, here you are as a scientist and you're wanting to produce some sort of new strain that's going to be produce more yield. So you take your existing strains and you're going to put them in conditions where horizontal transfer is likely to occur, and that's what the scientists will do. When it comes to fungi and yeast, when you're looking to produce new strains with those, then that can be brought about by sexual reproduction between existing strains. Because you've got sexual reproduction there, you've got the opportunity to be producing lots and lots of different variations there, and you can look and investigate to see which fungi or which yeast grows best in certain conditions and so on and so forth. So now you can have a wee look at your textbook, and on page 202 to page number 208, this information is there, and you can have a look at some of the testing your knowledge questions. And we'll look at recombinant DNA technology in the next video. Thanks for listening.